Our next presentation this morning is entitled Assessing Photosynthetic Response of Peanut at Different Planting Dates Using UAV-Based Multispectral Images. Chiara Rossi is our presenter. And Chiara, please turn your microphone on.
Sorry, thank you for the introduction, Dr. Cameron. I will share my screen. Can you see it?
Yes, it looks good.
Okay, thank you. So as you mentioned, my name is Chiara Rossi and I'm a PhD student from University of Georgia. Today I will present my project, which is about assessing photosynthetic response of peanuts to different planting dates using UAV-based multispectral images. I will go through the introduction, the objectives, and the methodology. Then I will discuss our results, conclusion, and at the end I will speak about future research. So physiological measurements are very important to identify, but also quantify the status of the plants during the whole season. As we all know, photosynthesis is the process that best explains the status of the plants. And from this graph, we can see that net photosynthesis is highly correlated with stomatal conductance, which is another important physiological parameter. In fact, the leaves are full of stomata, which are small pores that are involved in different processes. One of them is transpiration, that is involved the movement of water and nutrients around the plants, and also gas exchange between the leaves and the surrounding air. Also, by improving photosynthesis, the yields of the plants increase, and this is also very important. However, physiological parameters are influenced by some factors. One of them is the changes in the environmental conditions. In the coastal plain of the US, for example, where this experiment was conducted, peanuts are usually planted between mid-April and mid-June. And because of this wide range of time, the plants are subjected to different environmental conditions, so they respond in different ways, and so the physiological parameter can change. Another factor is that most of the physiological parameter has to be measured in a specific time frame. For example, to measure photosynthesis with a gas analyzer, which is an equipment, it's better to take the measurement around solar noon. Another factor is that most of the physiological parameter has to be collected and analyzed manually, which is very accurate, but also time consuming. And that's why recent studies have demonstrated that remote sensing can be used to correlate vegetation indices with physiological parameters, and that this method is really helpful, especially in a large scale field. So based on this, the objectives of this project were to identify vegetation indices from a UAV-based multispectral images and to correlate them with the physiological data that we collect in the field in order to identify the status of peanut plants. And our hypothesis was that vegetation indices will be highly correlated with the physiological parameters if the variability of the environmental conditions are greater. So this is our field. It's located in Tifton, Georgia, and is part of University of Georgia Research Centers. The experiment was conducted last year, and the cultivar used was Georgia 6G. As you can see, the field is divided in 3 blocks, each representing one planting date. From the left, we have the first planting date that was planted in April 30, the second one in May 21st, and the third one in June 4th. Every planting date is divided in 12 replications for a total of 36. So the first part of our measurements are physiological measurements. They started when the plants were 45 days after planting until 143. And all the measures were collected in the second open leaves because it has a higher photosynthetic rate. For the physiological measurement, we used 3 different equipment. The first one is the infrared gas analyzer, which measures gas exchange and fluorescence parameters. The second one was a portable fluorometer that measures chlorophyll A fluorescence. For this measurement, we use an OGIP analysis in a dark room. This analysis study the thylakoid reaction, which is a group of reactions that involve the photosystem 2 and the photosystem 1. Both photosystems absorb light, and they use the electrons to oxidate water and to create ATP. The third equipment was a spectrophotometer that was used to calculate the pigment content, so chlorophyll A, B, and total carotenoid. The second part of our measurements were UAV-based multispectral images that were obtained with a 3D R solo UAV connected to a Parrot Sequoia multispectral camera. All the images were calibrated in 2 different ways. The first one was radiometric calibration using a calibration panel for 4 different bands. So green, red, red edge, and near infrared. While the second calibration was the geometric calibration that was obtained using a GPS and ground control points located in the 4 edges of the field. So all the vegetation indices were calculated with different formulas that are shown in this slide. And all the data were analyzed using Pearson correlation with JUMP software. And regression lines were created in order to compare the determination coefficient and the root mean square error in order to identify the vegetation index that best correlate with the physiological parameter. So as I mentioned before, based on the days after planting, the plants are subjected to different environmental conditions. So we have total water and temperature. In this graph, I will show you these 2 parameters for the 3 planting dates. So for the first planting date, we can see that while the temperature is well distributed, the total water is not. So we have some peaks during the end of the first planting date. For the second planting date, instead, we can see that both temperature and total water are more or less well distributed. So since peak flowering until harvesting time. While for the third planting date, the 2 parameters are not well distributed. For total water, we have some peak at the beginning and at the end of the season, but very important, we can see that the values of temperature drops at the end of the third planting date. And these affect some of the physiological parameters, and I will show you some graphs related to this. So now I want to show you our results. This first part is related to physiological parameters. All the graphs are built in the same way. So in the x-axis, we have the days after planting, and on the y-axis, we have some physiological parameters. So on the left, we can see the graph related to chlorophyll A and B for the 3 planting dates. So we can see that there is some variability within the same planting date, but also between the 3 planting dates. On the right instead, we can see the graph related to quantum yield of the photosystem 2. In this case, we can see that the variability increased a little bit in the second and third planting date. Again, on the left, we can see the graph related to net photosynthesis. Also, in this case, the variability is higher in the second and third planting date. But very important, we can see that at the end of the third planting dates, the value for net photosynthesis decreased. And this, as I mentioned before, is related to the drops in temperature at the end of the third planting date. On the right, we can see the graph related to stomatal conductance. Also in this case, the variability is higher in the second and third planting date. And again, we can see a decrease in stomatal conductance values at the end of the third planting date. And this is because, as I mentioned at the beginning, stomatal conductance is highly correlated with net photosynthesis. So both graphs follow the same trend. After analyzing all the physiological parameters, we calculate the vegetation indices and we correlate them with the physiological parameter. For the first planting date, we had some significant correlations, but our R-square were low, so we didn't feel confident to show any result. For the second and third planting date, instead, we had many significant correlations, but since we couldn't show all of them in this presentation, we decided to create some graphs in order to identify the vegetation index that best correlate with the physiological parameter. This is just an example, and it's related to the third planting date, but we did the same thing for the second planting date. So these two graphs are built in the same way. In the x-axis, we have the vegetation indices, while in the y-axis, we have the R-square on the left, that is represented with gray bars. And on the right, we have the error that is represented with the red line. So the first graph on the left is related to stomatal conductance. And we can see that one of the best vegetation index is the nonlinear index, because the R-square is really high while the error is low. Another good vegetation index that we select was the simple ratio. On the right instead, we can see the graph for internal leaf CO2 concentration. And in this case, we selected the modified nonlinear index. So I will show you now some of the results. All of them are built in the same way. So we have the vegetation index on the x-axis, while on the y-axis we have the physiological parameter. Then for the both planting dates, so second and third. For the second planting date, we use a black color, while for the third one, a red color. So this first graph is about the CO2 concentration inside the leaf and the modified nonlinear index. We can see that both regression lines are positive. They follow the same trend. The R-square of the second planting date is moderate, while the R-square of the third planting date is higher. The second graph is related to the correlation between stomatal conductance and simple ratio. Also in this case, both regression lines are positive. They follow the same trend. And the R-square of the third planting date is higher of the one of the second planting date. The last graph represents the correlation between, again, stomatal conductance, but now a different vegetation index, the nonlinear index. And again, both regression lines are positive. They follow the same trend. And again, the third planting date has a higher R-square. And the reason why the R-square of the third planting date is always higher is because during the third planting date, the environmental condition had a wide range of variability. That's why our R-square is higher in the third planting date than in the second. So based on this, we can say that the vegetation indices were strongly correlated with stomatal conductance. That the three vegetation indices I showed in the graphs, so MNLI, SR, and NLI, have a greater potential to estimate the status of the plant in peanuts, and that these vegetation indices have a better performance in peanuts planted after mid-May. However, there are future research that can be done. One for sure is to validate the model that we obtained, so the correlation between vegetation indices and physiological parameters, but also to test the stability of this relationship. So maybe under different environmental conditions, cultivars, irrigation system, or soil type. With that, I would like to say thank you to my advisor, Dr. Cristiani Pilon, and the professor that participated in this project, Dr. George Belidis and Dr. Scott Tubbs. I would like to say thank you to the physiology team members and all the technicians, and at the end to Georgia Seed Development and National Peanut Board. Thank you so much for your attention, and I will accept any questions that you might have.
All right, thank you for this presentation. We have time for questions. Any questions right now? First question comes for you from Dr. Simerjeet Virk, and his question is, which method did you use to find the best fit line, linear, quadratic, etc., for the MNLI index and other parameters?
Can you repeat the question?
Yes. Which method did you use to find the best fit line, whether it's linear, quadratic, or other, for the MNLI index and your other parameters?
Okay, so the question is which method we use to identify our vegetation indices. We use the quadratic one.
Okay. And for now, that's the questions we have in the time. Thank you very much for your presentation.
Thank you.